So DNA replicates to form a new DNA. So that is the first point. And your body, you all know that your body is fully made up of proteins. So how these proteins are being produced in our body? There are numerous number of proteins, numerous number of enzymes in your body, and all these have to act in a precise and a coordinated manner. Okay, so how all these things are getting carried out, how all the things or such a complex thing are being produced in our body, and it is different for each and every individual. So how is this possible? This is only possible from the codes of DNA. Okay, so from the DNA, you derived a primary transcript. It is like copying of DNA from one format to another format. Okay, for example, it is like, how to say, you have a primary transcript. I mean, you have a primary material which is in a written format. Okay, and you are trying to digitalize that by typing. Okay, so it is just a change of format. There is no change in the main content. Okay, just the format is being changed. Okay, so from DNA, you produce a transcript that is called as messenger RNA. Okay, and this process is called as transcription. You are producing a transcript from the DNA. Okay, so this process is called transcription. Okay, so from this messenger RNA, you are trying to decode that messenger RNA, and by decoding that messenger RNA, you are trying to produce the specified protein which is being encoded in the DNA. So how do you call this? It is changing from one language to another language. It is changing from the language of DNA to the language of protein. So how do you call that? It is called as translation. So this is the central dogma of molecular biology. Okay, DNA which replicates itself to carry on all the genetic codes incorporated within it and pass it on to the progeny, offsprings, as well as to maintain all over the cells, all over the body. And from this, in order to produce a protein, you first need to change that format in a more presentable manner. So this changing of format from DNA to that of mRNA is called as transcription. So from mRNA, it is from the DNA or RNA language. From that language, you are basically changing that to the language of protein. So this is called as translation, guys. Okay. So now, what is the difference between replication and transcription? What do you think is the difference between replication and transcription? Replication is nothing but from DNA, you basically form another DNA. So DNA to DNA, a new DNA is being formed. Okay. Whereas in case of transcription, you form messenger RNA from DNA. You form what? Messenger RNA from DNA. That is called as transcription. As I told you before, it is just a change of format from a more authentic written format into a digital format. You got the point? So that is the difference between replication and transcription. Okay. So what it involves in replication, it is formation of new DNA. Okay. So what kind of nucleotides do you think will be required for replication? You need deoxynucleotides. Whereas in case of transcription, what kind of nucleotides do you think will be required? It just involves formation of RNA, messenger RNA. It is just an RNA. So it is just ribonucleotides. Okay. So what is the difference between deoxynucleotides and normal ribonucleotides? What is the difference? Deoxyribo and ribonucleotides. So the thing is, deoxyribonucleotides means you have got a removal of oxygen at the second position of that sugar molecule. Okay, so because of that removal of oxygen from the second position, it actually gives a more stable structure. And you want your DNA to be stable or not? Your DNA has to be stable because it carries all the genetic information for the generation. Okay, and this genetic information, you don't want those kind of genetic information to get altered or to get disintegrated or to get changed just like that. It has to maintain its integrity. Okay, so in order to maintain that integrity, you see deoxyribonucleotides gives a more stable configuration. And I think I have explained the structure-wise diagram for the same in the topic of replication. Okay, so I don't want to waste the time in this drawing the same thing again. Okay, so deoxyribonucleotides gives a more stable and a closed form, whereas ribonucleotides have a oxygen at the second position, and it is because of that presence, what happens, the ribonucleotides are open for various kinds of other disturbances, other external disturbances, and they may get degraded very quickly. Okay, so that is the main difference between the deoxyribonucleotides and ribonucleotides. Okay, so why replication involves a more stable structure? Because you don't want your genetic codes to get altered between each generation. You want that intact. Okay, you want that integrity of your genetic code to be passed on from generation to generations. Otherwise, the future generation won't sustain at all due to mutations.